Okay, we'll make a start and a few people will join as we go, I'm sure. So my name is Jan Strugnell and I'm the Director for the Centre for Sustainable Tropical Fisheries and Aquaculture, or the CSTFA, and I'd like to welcome you to our seminar series. So the CSTFA provides world-class multidisciplinary solutions-focused research for local, state, federal and international resource managers, both in government and the private sector. And for those of you that are joining us from overseas, we're based at James Cook Uni in Townsville in far north Queensland, Australia. And I encourage you to check out our website, or if you'd like to find out more, please send me an email. And today it's my great pleasure to welcome Professor Ross Houston to speak to us. Professor Houston leads a research program on aquaculture genetics at the Roslin Institute, with a focus on selective breeding and genome editing technologies in aquaculture species. His primary research interests include development and application of genomic tools for aquaculture species, including sequencing and functional annotation of genomes. He's also interested in genomic selection for commercially relevant traits in breeding programs, with a focus on disease resistance, and also optimizing and applying CRISPR-Cas9 genome editing in vitro and in vivo to pinpoint functional disease resistance alleles. Professor Houston has been amongst the first to develop key underpinning genomic tools for several major aquaculture species, for example, high-density single nucleotide polymorphism arrays for Atlantic salmon, Pacific oyster and Nile tilapia, as well as genotyping by sequencing approaches. He's also studied genomic selection for several fish and shellfish species, including studying genotype imputation approaches and to improve cost efficiency. His discovery of a major quantitative trait locus explaining almost all of the genetic variation in resistance to infectious pancreatic necrosis in salmon has been widely applied in the salmon breeding industry to control this disease. He has a leading role in the functional annotation of genomes of aquaculture species and is involved in major European projects to apply genomic tools to improve disease resistance. And he's led studies to optimise CRISPR-Cas9 genome editing in salmonid cell lines and embryos, and is applying these tools to understand and improve disease resistance. We're really grateful that he's agreed to give a webinar to the CSTFA today, and thank him very much for his time. If you have any questions as you go, I ask you to put them in the Q&A at the bottom of the screen, and we'll, at the end of Ross's talk, I'll unmute you so you can ask your questions. Over to you, Ross, and thank you so much.
Thank you very much, Jan. Can you hear me okay?
Yes.
Thank you for the introduction. I couldn't have written it better myself. It's very kind of you to have me here to give this seminar. And it's great to at least virtually meet some of you folks there at CSTFA, and I know about some of the great research that's going on there. So I'm just going to start by attempting to share my screen. No, I can't work out how to do it. Oh, yes, I can. Okay, so hopefully you can see my first slide.
Yes, that's great.
Okay, thank you very much. So I'm going to talk today about releasing the genetic potential of aquaculture species. What role for genomics? Now, I should mention for those that are not in the UK that it's 7 AM here, so you might hear in the background the probably quite familiar sound to some of you of kids getting ready for school. So if you hear screaming, then there's probably not much to worry about, but I'll try to make sure that it doesn't disrupt the talk too much. So sorry, slide's not changing. It's just delayed. Sorry about that. A bit of a delayed reaction with the slide. Okay, so we're based here, which is just south of Edinburgh, in a sort of semi-rural location. That photo is taken on just a typical random day. The weather's always like that. You can see we're sort of nestled at the foot of what's called the Pentland Hills. The building on the right there is the Roslin Institute. It was built in 2011. And to the left, we have other buildings on the campus, which include an innovation centre and the vet school of Edinburgh University. So we're all co-located there, and it's a sort of hub of animal science, one of the leading hubs in the UK. And traditionally, we've been focused on terrestrial animals, terrestrial livestock in particular. But increasingly, we have a big focus on aquaculture. And a lot of our research in aquaculture, we have an expanding group there that are focused on particularly on aquaculture genetics and aquaculture health, and particularly applied to selective breeding, as Jan said in the introduction. So much of the talk today is going to be about applied genetics in aquaculture, i.e. selective breeding. We're at a fairly exciting time. I hope I'm preaching to the converted here, but we're at a fairly exciting time for aquaculture genetics. The domestication of aquaculture species is recent, it's rapid, it's ongoing, and that means that there's huge potential for genetic improvement if it's done properly. You can see there on the figure on the right that in contrast to terrestrial livestock, where farming has been going on for thousands and generally up to 10,000 years, aquaculture is, for most species, much more recent. And in particular, the use of selective breeding, the use of genetic improvement technologies is also much more recent, and it's occurring in parallel to this domestication process. So that means that there's a lot of standard genetic variation for traits of interest, a lot of potential for genetic improvement. But there's challenges. So production is based on a highly numerous and very diverse set of species across the evolutionary tree, and you can also see that on the figure on the right in the light grey bar. So while 3 species make up approximately 80% of global livestock meat production, we require many more species to get to that total of aquaculture production. And that means that we really need tailored and specialized breeding programs according to the specifics of the species, the biology of the species, and likewise with the application of genomics tools, it needs to be tailored to the situation, and it certainly needs to be low cost, as we'll come on to later. Another thing that we as aquaculture geneticists have in our favor is that we have very high fecundity and external fertilization being a feature of most of aquaculture species. So even for the relatively less fecund species such as Nile tilapia, you can still get perhaps several hundred to 1,000 offspring from a single cross. And then as you go up through some of the marine fish species, you can get tens of thousands to perhaps even a million. With some of the shellfish species, bivalves, perhaps even tens of millions from a single cross, a single family. And that's really advantageous for breeding and genetics research, because it means we have an amenable structure for high resolution genetics experiments. It's possible to do specified crosses in a way and in numbers that's not necessarily possible for other species. And we, particularly with the trait of disease resistance, we can relatively easily do large-scale experimental disease challenges. So just briefly about the UK aquaculture scene, it's really dominated by Atlantic salmon. There was 190,000 tonnes produced last year, and we're the 3rd largest producer in the world, behind Norway and Chile. You can see there some of the majority of the salmon production sites there tend to be located in fairly wet and windy areas on the west coast and the islands of Scotland. We used to be able to say we were the top salmon producer in the EU, because Norway is not in the EU, but unfortunately, outcome of certain political decisions made mean that we can no longer make that claim, an unintended consequence, I suppose. And in terms of the selective breeding of Atlantic salmon, it's really up there amongst the most advanced in, at least in terms of aquaculture, but probably in terms of all livestock species, terrestrial or aquatic, in the world. And it's dominated by few large breeding companies that are, or at least breeding or consolidated breeding and production companies that produce the vast majority of the eggs for production. And just very simply, when we're looking at traits that form a major part of the breeding goal, which typically around 50% of the breeding goal is disease resistance, at least for Atlantic salmon. Of course, these traits are very difficult or in some cases impossible to measure on the selection candidates themselves, because in some cases they cause mortality when you measure the trait. But also, you don't want to compromise biosecurity by having pathogens or disease challenges anywhere near your breeding nucleus. So what's done is this process of SIB testing, where breeding values of candidates are estimated from the siblings of the selection candidates that are kept there in a biosecure nucleus. And for this example of one family, of which there'd be many in a breeding program, several hundred perhaps, you split the offspring into two groups or multiple groups, and one of the groups stays in the breeding nucleus, biosecure, and the other goes for sibling testing, so perhaps disease challenge experiment, and that's used to calculate family level breeding values, and can be used to decide which families to take forward for producing the next generation. And what's happened, at least since the turn of the century, is that we've now been using genetic markers as well as the more traditional pedigree information to capture within family genetic variation. So here you can see that although we can get an approximate estimate of relationship between any two individuals via the pedigree, that is really an average value, and it varies a lot around that average. And this example here shows the realized relationship for full siblings, and you can see its average is at 0.5, but there's a lot of variation round about it. And so how do we get at that? Well, there's two main ways of capitalizing on that within family genetic variation. The first is marker assisted selection, and that's suitable for situations where there's a major gene affecting the trait that you're interested in improving. For example, resistance to IPN virus, which I'll happily talk about in the next few slides. And also that in the case, which is the majority of traits, where actually, yes, they're heritable, but they're underpinned by many QTL of small effect, well, that's where genomic selection comes into its own. And I'll talk quite a bit about genomic selection and how it can be applied to aquaculture later in the presentation. So the curious case of infectious pancreatic necrosis virus. So this was a previously problematic viral disease of salmon fry and post-smolts. And a long time ago, both our group and a group led by Thomas Moen in Norway found that a single locus explained almost all of the genetic variation in resistance. And that's there on chromosome 26 of the salmon genome. And this is the sort of difference in mortality level we saw between animals carrying two resistant alleles or two susceptible alleles. A huge difference in mortality when either in a field outbreak or when they're challenged with the virus in an experimental challenge. And there's a dominant acting effect. So you can see that in the sort of Punnett square type diagram at the bottom, you can see that individuals only need to inherit one resistant allele to be protected from mortality. And that's resulted in widespread adoption of marker-assisted selection in all major salmon-producing countries, first using old-fashioned things called microsatellites, which work pretty well, and then SNPs, which also work equally well. And the result is, as you can see, that the reduction in salmon mortalities due to IPM was quite marked. This is just data from Marine Harvest, which is now called Maui. And the number of mortalities went from several million, tens of millions per year in Norway down to very few over the case of 5 years or so. But what it also did is something like this. We had a situation where there was very high expectations because everyone, or not everyone, but people thought that to tackle diseases with genetics, you just needed to find a gene, find a marker, and that would solve your problems. And when they looked, they found that, well, actually for most diseases and most traits, there's not actually any evidence for major QTL, so they're polygenic traits. And that's where we reach this plateau of productivity where genomic selection has really found its place. And genomic selection involves the use of genome-wide markers to predict breeding values for polygenic traits. And this is really an extension of the diagram I showed earlier, where we have a situation where there's many families, a couple of hundred families, say, within a breeding program, and you have reference populations with representation from all of the families in the breeding program. That reference population becomes the group of animals that can be sent out for SIB testing. So, for example, disease challenge. The selection candidates, again, are maintained in a biosecure breeding nucleus. And for the reference population, you collect genotypes using, for example, a high-density SNP array and disease phenotypes. And for the selection candidates, just genotypes only. The reference population allows you to train your genomic prediction model, and that allows you to make the selection of the best candidates, importantly, using genomic breeding values, which means it's both within and between families. And just to give an example of that, we've done some work looking at resistance to amoebic gill disease in Atlantic salmon. So this is probably at least one of the top 2 disease problems for Atlantic salmon. We collected data from a relatively large population of salmon from a commercial breeding program. They were measured for gill score and amoebic load following a challenge. And then we did a genome-wide association study and found, as I was alluding to earlier, a bit disappointing results that there's, yes, it's a heritable trait, of course, but there isn't any evidence for any major QTL, and there wasn't actually any QTL that's surpassed the genome-wide significance threshold. So here we're looking at a polygenic genetic architecture. So using the same data, we wanted to test or have some indication of how effective genomic selection is going to be. So we're actually testing genomic prediction. We're testing how well can we predict the resistance, or we want to test how well we can predict the resistance of broodstock without trait measurements. And so how we do that in a test scenario is we have this population of fish with genotypes and phenotypes, and we mask some of the phenotypes of a subset of the fish. And then we ask, using the genomic prediction model, how well can we predict the mass data using the genotypes only? And if we repeat that several times in a process known as cross-validation, we get an indication of correlation between the predicted and true trait values. Because what we're really looking at is trying to work out how well we can predict the true breeding values, we do an adjustment for heritability to get some indication of what the prediction accuracy really is. And so we did that for amoebic gill disease resistance in salmon, and we found that here you have SNP marker density on the x-axis and accuracy on the y-axis. And we found that for both traits, amoebic load and gill score, we have a significantly improved prediction accuracy with genomic selection compared to pedigree. So that's encouraging result number one, but encouraging result number two is that if you go, I don't know if you can see the cursor, but if you go down to a marker density even as low as 2,000 SNPs, well, there's not really much difference between 2,000 and 7,000, and indeed if we take that up to even 50,000 SNPs, we find that the accuracy does not further increase. So we've got this sort of asymptote of accuracy at 2,000 SNPs in this Atlantic salmon situation. So how repeatable is that across species and traits within aquaculture? Well, a while back we did a review of 36 studies in 18 species comparing pedigree and genomic prediction. And these were very diverse traits. They were diverse in terms of their SNP density, their sample size, the technology used to collect genotypes. There is a very clear pattern, and the median increase in accuracy using genomic prediction versus pedigree for growth-related traits was 24%, and disease was 22%. And so, of course, this could translate directly to genetic gain, and it could be transformative if, and this is the key point, if it can be widely implemented in many aquaculture species. And of course, most aquaculture species don't have the resources that Atlantic salmon have in terms of the R&D programs that can support the breeding programs. And so we have to think about, what about aquaculture species with fewer genomic tools? I mean, the typical technologies used for genotyping currently are genotyping by sequencing. For example, RAD sequencing has been widely applied of various iterations. And that's particularly suitable for generating genome-wide SNP data in perhaps new aquaculture species or smaller industries where perhaps generating a SNP array is not cost effective. However, having said that, SNP arrays are becoming cheaper, and that includes bespoke multi-species SNP arrays where you can reduce the costs by combining the SNPs onto one array, particularly suitable where the industries are fairly closely aligned. And we've got an example in Europe of the two major Mediterranean aquaculture species, European sea bass and gilt-head sea bream. We created a SNP array recently that combined approximately 35,000 SNPs per species onto the same array via these two European projects, MedAid and PerformFish. There was sampling done from throughout the Mediterranean, where most of the aquaculture of these species happens, and also a few wild populations. And then we selected SNPs distributed throughout the genome according to well, what you can see here is the similarity in pattern in track B of the Circos plot here between nucleotide diversity, which was used as a sort of proxy of recombination rate, and the density of SNPs in the SNP array, which is track C for the bass and bream, respectively. So that's one way to reduce costs, but you're still having 35,000 markers per species. Do you need that density? We did another study where we looked at the impact of SNP density on genomic prediction accuracy across several species and traits. And this was again comparing quite diverse species, quite diverse traits and genotyping technologies. But the pattern was actually quite consistent. So we found that even down at 2,000 SNPs, or perhaps even slightly less, we're still able to capture most of the genomic prediction accuracy that was seen with the full density panel. So that seems to be something of a general rule in these aquaculture species, at least where we have reasonable sized full SIB families within the training and validation set. And can we go further? So thinking about even more cost-effective genomic strategies. Genotype imputation is something we've worked a bit on. So here we're genotyping key individuals at high density, but the vast majority of the individuals in the population at low density. So in an aquaculture setting, the key individuals would typically be the parents, perhaps genotyped at high density. We need mapped, ordered SNPs. So of course here, a map of the genome is essential, and preferably a high quality reference genome. And then the offspring can be genotyped at low density and imputed to high density. And so you can see here the two parents represented with the two haplotypes for each of them. If we just have low density genotypes for some of the for the offspring, we can work out which of the parental haplotypes they're carrying, and therefore fill in the gaps between the genotype markers. It's a bit like a game of genetic Sudoku. The reason we want to do that, of course, is because low density panels are much cheaper than high density panels, and we're Scottish and we're always interested in saving money. And it's quite interesting to think of these multi-purpose added value SNP panels. So can we combine, we use SNPs in aquaculture for multiple purposes, for parentage assignment in some cases, for imputation-based GEBVs, as I'm mentioning above, QTL markers for marker-assisted selection if we have them, assessment of population structure, traceability. So can we think of a multi-purpose SNP army knife, which gives us all of these things in one panel? And this is something, of course, that's very much on the agenda, and there's some excellent research coming out of James Cook in this area. And this is a paper from earlier this year by Guppy et al., where targeted GBS technologies were used for this low-cost multi-purpose genotyping for black tiger shrimp. And we can see here the reason I think that we're seeing some of the results that I've been showing is because if you can see here, you've got the number of SNPs used in calculating the genomic relationship matrix on the x-axis, and the correlation of that genomic relationship matrix with a full marker set on the y-axis. And if you look at the number, which is roughly around, say, 2,000 is the number, where 2,000 or 3,000, maybe up to 4,000 is where you can see that you can capture the full, accurately capture the full genomic relationship matrix. And this genotyping can be done for these 4,000 SNPs at a cost of 15 Australian dollars per sample. So obviously, you folks over there share the same philosophy on making things cheap and saving money. But can we perhaps go further than that? So we did a study a year or two ago where we looked at optimizing imputation strategies for genomic selection. This was based on parents and offspring from an Atlantic salmon breeding program. The parents were genotyped for 5,000 genome-wide SNPs, and the offspring were genotyped for low-density subsets, or at least in silico genotypes. So they all had the full data set, but we masked the varying proportions of that data set, and then they were imputed. The offspring were imputed back up to 5,000 based on the haplotypes that were calculated for the parents. Here you see the results from the accuracy of the imputation. So pretty good. The x-axis is the density of the imputation panel used, and the y-axis is the accuracy. Sorry, this is probably quite confusing, but in this case, this is the accuracy of imputation. So how accurately are we recapturing the true genotypes of the 5,000 genome-wide SNPs? And generally, it's up to and above 0.9 once you get to, say, 700 SNPs in the low-density panel. And of course, I guess with genomic prediction, the proof is in the pudding to some extent, because we can then go and see, okay, using these imputed SNPs, how well can we predict the traits using the cross-validation approach I mentioned earlier. So the red line is the accuracy. So the dashed line is the accuracy with a full data set. So you can see there around 0.54 is the accuracy of the full data set, in which case the SNP density, the x-axis, is not relevant. The the red line is the accuracy with the imputed data set. So that's if we have the imputation panels of a density as shown on the x-axis, if we then impute them up to 5,000 SNPs and do genomic prediction, what's the accuracy of prediction? And you can see it doesn't really differ from the full data set, so that's quite encouraging. And the light gray is the accuracy if we just use the low density panel on their own. So if we didn't do imputation, basically. And you can see that's quite a bit lower. So that's the sort of benefit of imputation we get at this, using even these very low marker density panels. At least in this scenario, we can get maximal genomic prediction accuracy with as low as 200 SNPs, which is at least very encouraging for cost-effective genomic selection in Atlantic salmon. And that might apply also to other species, perhaps where SNP arrays are not yet cost-effective. So that's been quite a good news story so far. But it's important to point out that one key factor in determining the accuracy is the relationship between the training and validation population. Or if you like, in the context of a breeding program, the relationship between the reference population and the selection candidates. And here we have an example from common carp where we looked at resistance to koi herpes virus. There were four factorial crosses in the study, each comprising 10 sires and 5 dams. So we didn't know the family structure of these factorial of these groups of offspring until we did the genotyping. They were mixed, and then a subset of the offspring were challenged with koi herpes virus. It was a successful challenge experiment with the typical expected daily mortality profile. And then we used, in this case, RAD sequencing to genotype all individuals, and we got about 15,000 high-quality SNPs. And then we tested genomic prediction using cross-validation under 4 scenarios with varying relationships between the training and validation sets. So we did first the sort of standard approach of random allocation cross-validation, and the accuracy was, if we just focus for now on the RR BLUP results, which are not very different from the Bayesian approach, was 0.52. If we ensured that we had full siblings in both the training and validation set, we had a slightly higher accuracy of 0.56. If we ensured that we had at least half siblings in both the training and validation set, it was still pretty good, 0.53. But as soon as we removed the full and half siblings from the training and validation set, so that in the validation set there's no full or half sibling in the reference population, well, the accuracy dropped very dramatically. So that suggests that you really need close relatives in your training set, and from a practical point of view, perhaps sibling testing, phenotyping is still going to be required each generation if this is going to work well. We've done a bit more of a systematic test of this concept in Atlantic salmon, where we've been using the genomic relationship and removing individuals above a certain relationship threshold from the training and validation sets. And we see a sort of similar pattern. So we've got the relationship on the x-axis and the accuracy values on the y-axis. This is actually for sea lice resistance in Atlantic salmon. And if we move individuals if we ensure that there's no individuals with a relationship threshold above 0.4, then there doesn't seem to be much difference. But as soon as we go down to 0.3, the accuracy drops dramatically. So really, the full siblings seem to be very important in this context of getting reasonably high accuracies. So is there anything we can do about that? Can we improve prediction in more distant relatives or perhaps across generations. And I think here is where functional genomics comes into its own to some extent. So we can look at functional enrichment and improve methods of predicting functional variants. And here, high quality reference genomes with functional annotation is really important. It's important for two main reasons, at least from my standpoint as applied to aquaculture genetics. One is improved genomic selection via potentially functional enrichment, and the other is prioritization of targets for genome editing, which I'll get onto in a second. And there's a large project that's ongoing just now in Europe, which we're involved in, called Aquafang. And this is building on some of the initiatives in terrestrial farmed animals to improve the annotation of the genomes of 6 of the most common, or the species, 6 of the aquaculture species with the largest production volume and value in Europe. And so what this can, what we can do with these sorts of data is compare standard versus functionally enriched SNP panels, look at generating population scale whole genome sequences, perhaps using the imputation approach mentioned earlier, and assess whether if you enrich for SNPs that are theoretically more likely to be functional as opposed to random SNPs, SNP markers, does that lead to an improvement in prediction accuracy or an improvement in the persistency of that accuracy as the relationship between training and validation population gets more distant? And it's functional genomics tools are also very valuable for understanding the mechanisms underlying the genetic basis of the traits we're interested in. And here, of course, annotation of genomes is also key. So, for example, we've done several studies where we've looked at gene expression comparison of divergent genotypes. These are either selected based on QTL or EBVs in relevant tissues. For example, again, looking at amoebic gill disease resistance in Atlantic salmon, we've compared resistant and susceptible, or animals with high and low EBVs for resistance and susceptibility, using RNA sequencing of the gill and the head kidney. And if we can, and what we did is overlaid the functional genomic data together with whole genome sequencing of resistant and susceptible individuals to identify putative functional candidates within QTL regions, albeit the QTL are rather small in effect. And so we've picked out some interesting candidate genes for further functional validation. Going further than that, we have new technologies which could be very useful for looking at a higher resolution at this concept. So single cell RNA sequencing for cell type specific comparison of host response between resistant and susceptible animals. If we have a tissue where we're getting a mixed population of cells, but with single cell sequencing, we can separate those out and compare the gene expression profiles between certain cell types in resistant and susceptible individuals. This is an example from infectious salmon anemia virus challenged salmon head kidney cells, where on the left they've been clustered according to their gene expression pattern, each dot being a cell. And on the right, we can see differences in the signatures of gene expression response between the different cell types. And that includes the difference, importantly, between infected cells and uninfected cells, which I think is going to be very useful for picking out functional genes. And then the final topic I wanted to cover today is genome editing, which I think sits somewhere towards the end of this process of moving from genome-wide association studies and genomic data through to understanding the function of some of the genes underlying traits of interest to aquaculture. So the most commonly used technique, CRISPR-Cas9, allows targeted genomic changes in the germline of the species we're interested in. And it does that by making a double-stranded break in the genomic DNA that repairs via one of two mechanisms, either non-homologous end joining, where the DNA break is repaired, but with a excuse me, with an error, or homology-directed repair, where a template DNA is supplied with a specific sequence that can result in a more precise edit. And this is typically done in Atlantic salmon, at least, and most aquaculture species via microinjection of CRISPR-Cas9 at close to the 1 cell stage of the developing embryo. And a very well-known example and great example is the knockout of the DND gene to induce sterility in Atlantic salmon. And you can see there's also, so here you can see the ablation of the gonadal development due to knockout of the DND gene. And the reason that the salmon on the right is a funny color is because this team, led by Anna Vergilius, also use a tracer edit by concurrently knocking out SLC45A2 to create albino salmon, so they can enrich for potentially edited fish. I think when it comes to some of the work that we've been describing earlier, CRISPR forms part of the pipeline to shortlist functional variants impacting the target traits, a way to go from a very large number of variants to a much smaller one. So if we start with high-powered genome-wide association studies, move on to whole genome sequencing and functional annotation, gene expression comparison between alternate genotypes, including expression QTL. Then I think the next stage, at least for disease traits that we're interested in, could be targeted editing of the functional variants in cell culture models, which we're fortunate to have for some of the species we're interested in, albeit not all of them. And then moving from there to targeted editing of the functional variants in vivo, and hopefully all the while reducing the number of potential functional variants and genes from a large number to a small number. We also don't want to restrict ourselves to just thinking about naturally occurring genetic variation in the populations. If we could combine genetic mapping with genome-wide CRISPR screens, for example, in cell culture systems, or even in vivo, and the sort of concepts given in this diagram here, if we can take sort of forward and reverse genetics approaches, then we can potentially find disease resistance alleles and genes that both are involved in naturally occurring genetic variation and resistance, but also understand genes that perhaps don't have naturally occurring polymorphisms, but if they're perturbed via CRISPR-Cas9, they could still be valuable alleles for potential application in the future. So we've been working a bit over the past few years in developing genome editing, but particularly, well, both in vivo and in cell culture. I would say cell culture editing methods certainly need development in aquaculture, so we've been focusing on that. We've got a study where we used ribonucleoprotein CRISPR complexes to perform high efficiency editing in the cell lines of several salmonid species. So cell lines from Atlantic salmon, rainbow trout, and Chinook salmon. This was we tested both Cas9 and Cas12a. Cas9 gave higher efficiency than Cas12a, but Cas12a increases the number of potential target sites within the genomes of these species. And we've used this to successfully target gene knockout, which gives a really valuable tool for functional validation of specific genes in salmonids, at least on a low throughput approach. But thinking more about this high throughput genome-wide approach for CRISPR, the typical method uses lentivirus transduction. So we looked at whether that's possible within the salmonid cell lines. The salmonid cell lines are quite difficult to work with, but there is a Cas9 stable, and it's also a green fluorescence protein stable Chinook salmon embryo cell line that we worked on. And we used a lentivirus mediated CRISPR approach to knock out the GFP fairly successfully. And we also sequenced the edits to show that there's a high efficiency of deletion and knockout edits. So it seems lentivirus mediated CRISPR editing is highly efficient, at least in this cell line. It doesn't work as well in the other cell lines. And so the next stage we're looking at, or we have underway now, is genome-wide CRISPR screening, or a GECO approach for disease resistance. And here we've designed guide RNAs targeting the knockout of all known genes in the target genome. In this case, it's the Chinook salmon genome. We deliver the guides in a lentivirus vector, aiming at a dose of about 1 integration per cell in a very large cell culture, and then screen for a challenge with a viral infection or a pathogen of interest, screen for a marker of pathogen resistance, which might be a marker on the cell surface, or at the simplest level, it might be cell survival, and sequence and look for enrichment of the guide RNAs. And theoretically, that enrichment highlights the genes that are involved in interaction between the pathogen and the cell, and potentially involved in disease resistance. Those can be taken forward for downstream testing and potential application in vivo. So we've done or we're undertaking the genome-wide CRISPR knockout approach, and we've designed the libraries for Chinook and Atlantic salmon, focusing on the Chinook salmon first, because that's where the lentivirus approach is working well. And this is the number of guide RNAs we can have. So you see we have a very large number of guide RNAs. Mostly we have 2 guide RNAs per gene in the Chinook salmon genome. Salmonids don't make life easy because they tend to have 2 copies of each gene due to the relatively recent whole genome duplication. And in that case, we've generally tried to design genes that can cut both copies of the paralogs. So just wrapping up, finally, what are the potential applications for genome editing and aquaculture breeding? I don't think we're quite there yet, but there's certainly a lot of potential. And I think it falls into 3 main categories. The first is we can use genome editing to increase the frequency or fix desirable alleles at existing QTL. That could occur also via selective breeding and genomic selection, but potentially genome editing can make it faster. The second and perhaps more exciting area is this process of potentially moving favorable alleles from one population or one strain or one species to another, or if you like, introgression by editing. And a good example here would be if a particular gene or set of genes that were responsible for the fairly well-known complete resistance of coho salmon to sea lice could be identified and modulated or moved to the Atlantic salmon, that could be transformative in terms of tackling one of the biggest barriers at the moment to Atlantic salmon production. And there's a large research project about to get underway on that topic. And the third is just thinking about this concept of not restricting to variation that occurs naturally in the population, but perhaps developing de novo mutations or alleles with desirable effects based on the knowledge of the biology of the trait. And that could also come from genome-wide screens. And a good example here is resistance to PRRS virus, porcine respiratory and reproductive syndrome virus in pigs. And a team based at Roslin, led by Christine Burkhardt et al., were able to make pigs that were completely resistant to PRRS, and that occurred by just knocking out a single exon, which encoded for a particular domain of the receptor CD163 for this disease, for this virus, and that made them completely resistant. So that's not a naturally occurring allele, that's a de novo allele, but it has this massive impact on resistance to the virus. So just to finish up, as I said at the start, I'm hopefully preaching to the converted here, but aquaculture species have major potential for genetic improvement, and this is really critical for global food production. I think it's one of the really exciting potential answers to some of the food security problems, where at this early stage in domestication, there's a high variation in selective breeding technology across the species. We do have the high fecundity and external fertilization, which allows for creative breeding designs and experimental approaches. but the use of many different species with very diverse biology necessitates tailored and certainly cost-efficient approaches to doing this. Genomic tools have been well demonstrated now can improve selection accuracy and genetic gain. This genomic selection and marker-assisted selection is a mature technology now in salmon, but the benefit has been shown in studies of at least 18 aquaculture species. So the question is how to translate those benefits that have been seen in the more advanced sectors to the emerging sectors. I think genotyping strategies are key, so thinking about cost-effective strategies, genotype imputation, multi-purpose panels, added value panels of SNPs. Trait data and close relatives appears to still be required, but perhaps functional enrichment might help with improving the persistency of selection accuracy in more distant relatives. Genome editing at the moment is a major research tool, and it has potential for production. We consider it part of the pipeline to discover functional variants for existing QTL, but also genome-wide CRISPR screens hold potential for looking at disease resistance, and the applications of editing can occur in several different ways, depending on the regulatory and public perception environment, of course, but perhaps fixation of segregating alleles, introgression of favorable alleles by editing, and creating new, completely de novo favorable alleles. And that's all from me. I'd like to thank all these people and more who've contributed to this work that I've presented today, which has been probably something of a whistle-stop tour of some of the research we've got going on. You can see there in the bottom right the members of my team, and I'd like to thank all of them for their contribution to this work. And thank you very much for listening.
Thanks so much, Ross. That was really interesting and yeah, really fascinating research. I was wondering if anyone has any questions, if you can make that clear in the Q&A, and I'll allow you to ask your question there. I'll just keep an eye on that while you're thinking about them. Ross, I was wondering, this might be a bit of a naive question, but I was wondering if what you thought about the utility of CRISPR-Cas9 for invertebrate aquaculture species, if that's likely to have any role, and are there technical reasons why not? I just am not aware of much going on in that space, and I was wondering what you thought about it.
Yeah, it's a great question, Jan, and I think the answer is obviously, well, to me, obviously yes, but there we're, the community, I guess, is facing the challenge of the enormous diversity of biology and the differences between the species. So we have some work ongoing at the moment looking at developing CRISPR approaches to look at disease resistance in Pacific oyster, for example. And of course, we're looking at new ways of delivering the CRISPR-Cas9, because practically, this idea of microinjecting at the one cell stage of embryos is not likely to be an efficient approach. So we probably need higher throughput approaches to delivering the CRISPR-Cas9 reagents. But certainly in principle, I think I see no reason. I see no reason why the same potential benefits couldn't be there for the invertebrates as well. I'm less familiar with the prawn and shrimp world, but I know there's been not a huge amount of publication in that area yet, and I think that's probably due to just again the diverse biology and technical issues. But yeah, I think it's got the potential. It's just getting the techniques to the stage, and that's the hard part, I guess, getting the techniques to the stage where they can be easily applied for the interesting research questions.
Yeah, thank you. I see Dean Jerry has a question there. I'll just unmute Dean so he can ask his question.
Hi Ross, good talk mate, thoroughly enjoyed it. My question is related to genomic selection. it's been put forward as a real disruptive technology to how we will conduct breeding programs in the future. And I just would like to hear your opinion on whether we're really seeing this disruption occur in aquaculture breeding programs outside of maybe salmon at the industrial level.
Yeah, thanks, Dean. Yeah, it's a great question. I mean, selective breeding per se is expensive, and genomic selection, I guess, even more so. And genomic selection is, particularly suitable to where there's already an advanced, perhaps family-based breeding program, and the level of selective breeding technology is highly variable across the different aquaculture species. So you're right. I think salmon kind of leads the way. I think we're now seeing other species follow suit. So Nile tilapia, shrimp, and perhaps rainbow trout, and some of the species I mentioned, like sea bass and sea bream, they all have genomic selection underway to some extent. I think in terms of making it affordable, let's say, to a sector that's just getting off the ground, or is perhaps smaller than salmon, I think that's where some of the approaches that, well, for example, that your team have been applying, where you look for, how do you get the added value in doing genotyping. So if you need genotyping for your family assignment, for example, or if you need a SNP panel for detecting population structure, then it's probably not that much more expensive to generate the data that you'd require, at least for the genomic breeding values, leaving aside, of course, the phenotyping costs. But I think that's where it can actually be, it can actually be quite affordable in terms of the genomic side.
Okay, thank you.
Are there any more questions for Ross?
Do I have a follow-up one, Jan?
Oh, okay, Dean, you can have a follow-up question, and then we'll have a question from Alexander Chong. So shoot, Dean.
AI as a way to improve prediction of our genomic selection models. Have you had any exposure with using AI as a training platform? And again, what do you think the potential of this might be to etch out even those higher accuracies that we might be able to find using genomics?
I actually don't have any experience of AI approaches. I think it clearly has some potential, particularly when it comes down to some of this, the idea of if you're starting with whole genome sequence population scale data, which probably that will eventually become norm for the aquaculture populations, then yeah, perhaps that AI approach for selecting the variants could be useful. But yeah, it's not something that we're actively working on, but I think it's certainly an interesting area. Yeah.
Okay, we have another question here from Alexander Chong. I've just, you should be able to ask your own question now, Alexander.
All right, thank you. Hi, Ross. Thank you for imparting your knowledge and ideas. It has been suggested that tropical species have a much higher genetic variation than temperate species. If this is true from your experience, will this facilitate the discovery of more beneficial traits in tropical farm species? Thank you.
Thank you, Alexander. I mean, I think I don't see that for the majority of aquaculture species, tropical or temperate, that lack of genetic variation is an issue. I think having the recent domestication and the the farming of species that are not so far from wild counterparts means that there is quite a lot of standing genetic variation in tropical species and probably also in temperate species. Of course, the key is management of the genetic diversity once you move to a situation where you, well, first of all, in selecting the animals for the base population of the breeding program, and then move to a situation where you're careful not to make mistakes in the selective breeding program that reduces the genetic diversity, because that obviously has negative consequences. But I think it's a general theme that we have access to these high levels of genetic diversity because we're at this early stage in the domestication process.
Okay, thank you.
Thank you.
Okay, so if there are no further questions, I'd just like to thank you, Ross, for getting up so early. I realised that was 7 o'clock at your time, so I think we really appreciate your efforts there, and should let you go and get some coffee into your system. But thanks so much for sharing your knowledge with us. It's been a real treat for us to hear from someone else interested in aquaculture genetics. So thanks so much for your time.
Thank you very much, Jan, and it's very kind of you to ask me to give the presentation, and thank you to everyone that sat through it. I even see some UK names on the attendance list, so some people obviously got up early enough to watch, which is great. But thanks to everyone for coming along and also for the good questions.
Thanks, Ross.